It is already after 3 p.m. and today we are scheduled to do our online lecture. Many people have not yet turned up for our revision because today is our revision. Many people have not turned up for the revision, but we fixed today as our revision week. Last week was our week 6. We did the 6th facilitation last week. And I promised you last week that today will be our revision class, so that we'll revise all the things we did from week 1 through week 2, through week 3, through week 4, through week 5, through week 6. So we will do all the things that we do from week 1. So today we want to revise what we did in week 1. We did introduction. We did introduction to evolution. We did introduction to evolution in week 1, where we define evolution. We say that evolution is changes that takes place in organisms from time over a long period of time. That's what we did in week 1. We introduced evolution. We also looked at theories of evolution. A lot of theories that were put in place to explain evolution. A lot of theories were there to explain evolution. We look at Darwin's theory, Jean Lamarck's theory, and other theories to explain evolution. We did that. Also, we look at the history of evolution. What was initially thought to be evolution, how people view evolution, a lot of theories that were put in place to explain evolution. In this week 1, we also looked at Hadley-Weinberg equilibrium, or what we call Hadley-Weinberg principles, or Hadley-Weinberg theory, which states that the genetic frequency of the allelic frequency of a population remains constant over a long period of time, provided that certain conditions are constant, that the allelic frequency of a population remains constant over a long period of time, provided that certain principles remain constant. What are those things that remain constant? Something like mutation, there will be no mutation. Then there will be no emigration or immigration. And then the population is large. There is no reshuffling of genes. These are some of the things that will remain constant for this Hadley-Weinberg theory to be truthful, that that this principle have to remain constant, that all the population, there will be no mutation, and then that there will be no migration, there will be no migration, there will be no immigration. That this thing has to be constant for the allele frequency to be in a population, the allele frequency in a population to remain constant, that these conditions have to be there. So we looked at that. That means we looked at the Hadwenberg equilibrium. And then we also looked at polymorphism. Poly means many, the existence of many forms of an organism. We looked at that as in week 1. In week 2, we looked at heredity and genetic variation. So in week 2, we explain what we mean by heredity. We explain what we mean by variation. And we say that heredity is the tendency of young ones to resemble each other and resemble their parents. Then we also mention variation, that the variation is tendency for the organism to be different from one another and to be different from their parents. Karim Mats, you are welcome. Can you say hello to us?
Good afternoon, sir.
Afternoon. What may you people, you mean you forgot about our revision today?
Sir, you tried to join through one link like that, but I was saying the host is not available. Not until you sent another link.
Okay, through your WhatsApp group. Huh?
Yes, sir.
Okay, okay. I hope others will also copy the WhatsApp group.
I will send the link to others now.
Do so. So we are doing a revision. We have said what we did in week 1. We have entered week 2. What we did in week 2. We did heredity, which is tendency of the offspring to resemble each other and resemble their parents. We also looked at a variation, which is the tendency of organism to be different from one another and also their parents. So we also looked at mutations. Mutation. We also did mutations. We said that mutation is the change in the arrangement of genes on the chromosomes, because the genes are arranged on the chromosomes. If, for example, the desk, the long bench that students sit on is a chromosome, then assuming that 6 people are sitting on that bench, each person is sitting on a specific position. So the position of a gene on a chromosome is called what? A locus. So each gene is staying on a locus of a chromosome because the genes are inside that chromosome, and there is a specific position that this gene stay in the chromosome, and that position is called locus. So when there is a change in the arrangement of gene, that means the gene move from maybe locus 1 to locus 6. Gene that's supposed to be in locus 1 have been pushed to locus 6. Then mutation is said to occur. And mutation can lead to a lot of problems. A lot of things can arise if mutation takes place. Then we have different types of mutation. We have chromosomal mutation and gene mutation. We looked at that in our week 2. Then we also looked at polyploidy, the process of having more than 2 sets of chromosomes. So it's polyploidy, and mainly in plants, even in some animals. Polyploidy is important in some plants because it helps to produce plants with large leaves, with large fruits, with all these improved crops we are seeing is as a result of inducement of polyploidy, so that the number of chromosomes becomes plenty. And when the number of chromosomes are increased, then increases the fruit, the size of the fruit, the size of the leaves, and even the other parts of plants. Also in animals, it can also happen in animals. Then we also look at isolation. When an organism is separated from other population, then you say it is isolation. And a lot of factors can lead to isolation. It may be geographical isolation or physiological isolation. Geographical isolation is that it is external, things around in the environment are separated the organism. It may be physiological, things inside the organisms are separated that organism. So has made the organism different from others. So that is what we call isolation. So what are the environmental factors that can lead to isolation? Something like hail, mountain, forest, ocean, rivers, deserts. All these things are environmental factors that can lead to isolation. That was what we did in week 2. In week 3, we did the origin of life. And then we looked at these theories, Darwin's theories of organic. We looked at organic evolution. Of course, we have organic. There is theory to show that life came into this world are of two types. There is theory of organic evolution and theory of divine creation. We are looking at how organism came to the earth. Some people propose that organism came to the earth through organic molecules. That is organic evolution. Others believe that organism came to the earth through a God. That is theory of divine creation. So these are the two theories to explain how life came to this world. How me and you came to this world. Some people believe that organism came to the world through organic molecules. And so if you believe that life came from organic molecules, then you are believing the theory of organic evolution. Then if you believe that God created you, that is God that made everything possible in our lives, then you are believing in theory of divine creation. Now, we can, if you go to organic evolution, which is the theory that says that life came from organic molecules, they have some evidences to support their position. And this theory of organic evolution is called scientific theory. That it is scientific because it is organic molecules are scientific materials. So the organic evolution, people that believe in organic evolution have evidences to support their belief. So evidences of organic evolution. There is embryological evidence to show that organic evolution is rare. They tell you that embryos of invertebrates and the embryos of vertebrates are all the same at the initial stage. And it is true. If you look at the embryo of, let's say, a fish, the embryo of a frog, the embryo of a snake, the embryo of a bird, the embryo of a monkey, the embryo of a pigeon, the embryo of a chimpanzee, the embryo of a human being, all of them are similar at the initial stage. So this is embryological evidence of evolution, of organic evolution, to tell you that embryos are all the same at the initial stage. And that is correct. The another evidence is anatomical evidence. They believe that another evidence of organic evolution is anatomical evidence. When you look at the fingers of man, they are 5. That is pentadactyl digits. When you look at the finger of a dog, they are 5. When you look at the finger of a monkey, they are 5. When you look at the fingers of a chimpanzee, they are 5, showing you that anatomical evidence is the evidence of organic evolution. Then they also have what we call evidence from vestigial organs. Vestigial organs are organs that are no longer in use. We call them vestigial organs. When you look at the vestigial organs, you find out that the other animals that are not man have that vestigial organs. But man, our own vestigial organ has been decreased or diminished. So, but we have it like those animals. Something like muscles of the forehead. If you look at a lion's face is very fearful because there's muscles in the face that makes it to be fearful. So also we have that muscles in our face as human beings, but it has diminished. It's no longer as prominent as that of a lion. And that's why when some people become annoyed, if you see their face, you run away. Some people have wrinkled face. If you watch it, it's true. So these are vestigial organs. Then appendix. Appendix is to show you that our stomach was like that of a goat that has 3 chambers. But because we are not goats, the thing has to reduce and remain in your intestine to form appendix to tell you that we are like a goat before. So another evidence to support organic evolution is paleontological evidence, or what we call fossil evidence, paleontology or fossil, something that we dig out from the soil. So a lot of things have been dig out from the soil, from the early man, showing you that we are like them before. So that is paleontology or fossil evidence. Then you have evidence from DNA or biochemical evidence. We have been able to isolate the DNA of some of these lower animals or even lower vertebrates and compare it with the vertebrates. They are similar. So DNA is a principal evidence to support organic evolution. Evidence from biochemistry, the amino acid sequence of these lower animals, when you compare it with the amino acid sequence of a man, you find out that they are almost the same. Then in week 3, we also did what we call adaptation. Of course, adaptation is a characteristic of living things that helps organisms to survive. Otherwise, if you move from Nigeria to London, you will not be able to survive because the weather there is different from our own. So, but because of adaptation, we're able to survive. Even if we move to London, even if we move to Ireland, even if we move to China, even if we come to Canada and back to Africa, the weather changes, but because you can adapt using different methods, then you'll be able to survive. In week 4, we explained polyploidy in detailed study of polyploidy. We did it in week 4. And then the origin, types of polyploidy, we did it in the week 4. Importance of polyploidy. Polyploidy in plants and animals. In week 4, that was when we explained it very well. In week 5, we look at adaptation more and speciation. We explained it, we defined it, and even the importance of adaptation, which I have said, because it enables organisms to survive, enables organisms, even new species can arise, and then it helps organisms to survive. Then we also classified, we also look at phylogeny. There's something we call phylogeny, there's something we call autogeny. Autogeny is the development of an organism is called what? Autogeny. Then phylogeny, the life history of an organism. When organism, during the pregnancy period, or when the organism is being formed, it tries to repeat the life history of these ancestors. So the life history of ancestors is called what? Phylogeny. While the development is called what? Autogeny. And that's why in evolution, we have what we call biogenetic law. Biogenetic law states that autogeny recapitulates what? Phylogeny. That is, an organism in its sequence of development repeat the life history of its ancestors. So development is autogeny, while phylogeny is what? The life history. It resembles. That's why in evolution, we can isolate the gene and then do a kind of phylogenetic tree, the relationship, how the organisms are related. So that's what we did in week 5. In week 6, we did what we call geographical isolation, which we have done. We also treated gametes and zygotes. Gametes are the sex cells, which are the male gametes and female gametes. The male gamete is the sperm, while the female gamete is the egg. The sperm are formed during the process of spermatogenesis. For example, when a child is born, it doesn't produce sperm. That male child is born, it doesn't produce sperm. That male child will grow. He has the germinal cells. He has these potential cells that will produce sperm, but it does not produce until that child reaches the age of 13. Then spermatogenesis will take place, a kind of creation of cells. That cells will be what? Made, will be produced. That germ cells originally put in place there. That's why when a child is born, he has penis, he has scrotum, but the sperm has not started forming. It's only when he reaches that stage of maturity that spermatogenesis will take place. Sperms are generated or created so that the individual will produce the sperm. And the egg, once a girl is born, it doesn't start ovulation and it doesn't start menstruation. Until that child reaches the age of 13 or 14, menstruation will now come, showing you that that girl is now mature. So an ovum is a gamete, a sperm is a gamete. And when the male gamete called sperm fertilizes the female gamete called the ovum, then a zygote is formed. And that zygote contains full complement of chromosomes because a sperm contains half the chromosome number as are present in the body cells. The ovum contains half the number of chromosomes as are present in other body cells. So the chromosomes in the sex cells are always half. So that when it meets the partner, when the sperm meets the egg, the half in the sperm and the half in the egg will meet to form a zygote that now contains the complement, the full complement, that's the diploid number of chromosomes. The former one was haploid. This sperm is carrying haploid, the egg is carrying haploid. When haploid meets haploid, they form diploid number of chromosomes. Then we also treated mortality in week 6. And we said that mortality is the tendency of organisms to die. The death of organisms. The death of organisms in a population is mortality. Certain factors can induce mortality, something like disease, something like hunger. These are the factors that can make mortality possible in a population. Then we also have natality. Natality is the number of young ones, the number of fertile eggs that are produced in a population. Mortality should never be higher than natality. If mortality is higher than natality, then that population is in a decreasing form. That population cannot grow. For example, in your village, if the number of people dying are greater than the number of people that are born, then your village is bound to doom. That village is not growing. The population is not growing. That means the population is declining, and nobody's praised for that. In some countries of the world, like in Germany, the population is declining. The population in what's that country? Is declining. Is in Europe. Very, very going down. And so because the mortality becomes higher than the natality, and that's why they are now encouraging people to immigrate to their country. They do a lot of promo to enable females to begin to born children again. Australia, the population is decreasing seriously in Australia. And that's why they are doing a lot of palliatives, a lot of promo to enable females to continue to born children. Now, we also looked at types of isolation: reproductive isolation, ethological isolation, physiological isolation, cytological isolation. These are what we did in week 6. We also look at chromosome numbers in plants and chromosome numbers in animals. Here also, we also looked a little bit on, we also studied The chromosome in different organisms, like in different plants, like the domestic oats, the peanuts, the sugarcane, banana, white potato, tobacco, apple, cotton. One thing you should know here is that polyploidy has taken place. The chromosome sets have been increased. All these are a kind of modification so that the plants that are producing becomes better than the original form. But then it goes with disadvantage. When in this polyploidy, what happens is that most of them are sterile. Most become sterile. They cannot produce other ones. And then most are not stable. In other words, you cannot use the seeds that are produced to produce another one. So that is the shortcoming of high number of chromosomes. That is shortcoming of polyploidy. These are the things that we did for you, starting from week 1, week 2, week 3, week 4, week 5, and week 6. I want you to ask questions so that I can listen to your side. I've been able to do 7 weeks of facilitation for you. This 7th week is revision for you. So if you have questions, you may ask.
Thank you, sir. God bless you, sir.
Karimat, thank you very much for joining very well. I'm happy for
You're
you.
welcome. Thank you, sir.
Yeah. If you have questions,
Sir,
if you don't have question, no problem.
all right, sir. Thanks for the summary.
Yeah.
But the material is broken.
Eh?
The material.
I will still send it now, as usual. I hope you got that of
Sir,
week 6.
yes, but what I'm concerned about is
The,
though the material I
the
collected,
PowerPoint.
the one I collected from the school is 226 pages.
You see?
The material I collected from my study center, it has 226 pages.
No, we break it. Like all this breaking that we did, forget about the pages. If you go to chapter 1, I hope you understand. Look at chapter 1 and look at what we did in week 1 and week 2. Just follow it
Okay.
chapter by chapter. So if you are reading it, all of that like 200 and so, it will fear you. But do it chapter by chapter, vis-a-vis what we have been able to do.
Right.
Yeah. Are you okay?
Thank you, sir. All right, sir. Thank you, sir.
Okay.
Yes, sir.
Very great. Thank you. Maybe I call it a day here. Is that okay?
Yes, sir.
Okay. So read very well. So don't just look at what you collected and begin to. So when you bring week 1, find out what we did here from week 1. When you go to week 2, also look at it. Do you understand? As you read, you link it up with what you are doing. What I will do later is to pull all our lectures from week 1 to week 6. I'll still post it so that in case you miss anyone, you can pick it and you listen to it. Is that okay?
Sir, that will be better. So I the one that will set our.
You say?
I the one that will set our.
I don't know. Karimat, you want to get everything from me. What is it? How do you pronounce your name?
Karimat.
Okay, Karimat. It resembles one
Yes.
Indian name I know. There's one Indian friend I know. His name is Karim.
Yes, the for a female is Karimat, for a male is Karim.
I have a friend from India. Okay, thank you very much. Okay. Bye.
Have a good day, sir.
Amen. Thank you.